I'm Jenny Pandol, and I'm here with Adriana Ponzanelli, and we are interviewing Dr. Sabine Hazan today, and just wanted to introduce who she is. So she is a triple board certified physician in gastroenterology, hepatology, and internal medicine. She's also the founder and CEO of Ventura Clinical Trials, and she's been doing clinical trials on GI diseases for about 22 years. She's also the founder of Progenobiome, which is a company focused on microbiome research. She's doing really cutting edge stuff, so we're excited to hear about that. And then she's also the author of her first book. It's called Let's Talk Shit. And she covers a lot of data. She's actually famous for her quote. She often says, "Show me the data." That's how we know Sabine is in the house. And, there you go. That's her book.
Where is yours, Jenny?
Oh yeah, my copy too. So I read this. I didn't know this, but she is the first woman ever accepted into the first woman accepted to University of Florida as a clinical gastroenterology fellow. So that's pretty cool. So like I said, she's a pioneer in the GI microbiome field. She's been doing fecal matter transplants for primarily for C. diff patients, and has had extremely good success rates. But her clinical trials with the FMT she's doing on various diseases, including autism, Crohn's. Sounds like she's getting into other neurological diseases as well. And so, yeah, she's going to give us a little update on what she's been doing the last couple of years. I think I met her about 3 or 4 years ago when she started really getting excited about this stuff. And so she's going to give us a little synopsis about that. And let's see, is there anything else that we need to tell the audience?
That we met her together and that we went to her clinic.
Okay, yeah. So the three of us got to all hang out together, and yeah, for her conference. She does a yearly conference, Microbiome, Malibu, Microbiome,
Ventura.
No, it's called Malibu Microbiome Conference. Actually, I don't know the exact title, but anyways, that's a yearly conference. We've attended a couple of those together, so that's been fun. And we always learn something at those conferences, because it's all MDs and researchers doing really cutting edge research. So yeah, that's what we're going to talk about with Dr. Sabine today. And yeah, she's full of energy. So enjoy.
Thank you, Jenny.
Thank you.
I tend to not say healthy anymore, healthy diet, because what is a healthy diet, right?
Mm-hmm.
Some people, a healthy diet is McDonald's French fries, because they're healthy and they live till whatever. So, you can't really, I think we have to do more precision medicine to understand the precise diet for the precise person and the precise race and the precise genetics. And so, for my oldest one, I was eating salads and lettuce, and my oldest one was craving. That's all she eats: lettuce, salads, etc. And then my youngest one, I was craving McDonald's French fries, nachos, pizza, all the junk food. And guess what? Super healthy. That's all she eats as well. And here we are. Here's two girls, right? And you would think, well, my oldest one is so healthy, she's eating so healthy. But actually, so is the little one. She's eating McDonald's French fries, and God help me for trying to stop that behavior, because she will say to me, "But Mom, my microbiome wants it." So what am I going to say? You're right. Your microbiome wants it. And, she's super slim. The girl's like very slim, healthy. And so she has a diverse microbiome. So what am I going to say? I can't really justify. If she was diabetic or if she was something, I would say, well, look, you're diabetic because of this, but I can't even say that to her. So, and people say, well, it's going to catch up to her. Maybe it's going to catch up when she changes her microbiome, when she takes those antibiotics that kills her gut. Maybe when she takes those medications for in vitro to get pregnant. I mean, who knows, trauma, stress, all that's going to change. But right now she's fine. God bless her. She's eating McDonald's and nachos and not gaining weight. Hey, I wish I could do that. So
that is so interesting because, do you remember probably 1970s, there was this man in the news, and he was eating an airplane. Do you remember this man who ate glass? So this
right.
man supposedly could eat glass and could
Right.
eat metals and could eat like the steel, and he was ready to eat an airplane. So I was kind of just researching if it was true, and I don't know if it's true or not, but that was a kind of an idea I had, like, how could this man live? I think he passed away at his 50s, but how can this man, like, I can't, there was a point I couldn't even eat celery. So how can this man, eat like this? And I can't.
Well, I mean, obviously he's got a strong microbiome that does that. So,
yeah.
I mean, you see people that, survive COVID, especially we're in the era of COVID now, can't even do anything without opening the news. It's COVID is everywhere. And, you see these people that eat a horrible diet and they're exposed to their wives that have COVID and they don't catch it.
Mm-hmm.
What happens there? And you're thinking, well, but then vice versa, you've got these guys, these people that are exposed and that have a horrible diet, that are diabetic, that are overweight, and then they catch COVID and they don't do so well. So there's always these exclusions. I think, immunity is in the gut, in my opinion, and we have to start looking at the gut and we have to start paying attention of what we're putting in our guts.
Yeah.
And so that's why I'm on this big movement now of, making sure everything is checked, because, from the yogurt you're eating, we have to push the agencies to start monitoring better, to have somebody that overlooks these probiotics, these vitamins. When COVID hit, the first thing I went to CVS and I asked myself, is it real vitamin C? Who's making it? Where did it come from? Because if it came from China and they had COVID there, could it be contaminated? So, maybe that, I want to be overlooking the lab. I want to overlook everything, including the farming. I was just giving a lecture at a farm, and it's a regenerative farm with Joel Salatin, who's actually a famous farmer. And the first thing that came out of his mouth was, I believe in diversity of plants. I believe in diversity of animals. I believe in diversity. And, my whole movement and my discovery with the microbiome is really diversity equals health. Loss of diversity, and you've heard me on lectures, loss of diversity equals disease, right? That was our big finding, when we showed loss of diversity in Crohn's disease, that was a big finding. When we showed loss of diversity in Clostridium difficile patients, patients that got C. diff and then almost died, it was not because they went to the hospital and they were exposed to a nurse that gave them C. diff like we used to think. That's old mentality. It was because we gave them those antibiotics that killed their diversity, their gut flora, and therefore allowed C. diff to stimulate the toxin which basically caused, the C. diff toxin to secrete, and then ended up in the hospital. So I think diversity is very important. I think, when you look at kids with autism, loss of diversity. And so when you think of all that, it makes sense that if your cows don't have a diverse microbiome, if your food is not diversified, you're getting yourself into an area of problems. And, as much as we want to push a certain probiotic or a certain food, it may not be the right thing for everyone, right? So, beans are good for some people, not good for me. I can't eat them, lima beans especially. So, I think we have to rethink the whole nutrition and really make it more specific to the culture, specific to the region. And I think that's really my path, is really keeping those diversity, keeping that beautiful, difference between all the races. Where you go to Morocco and people are eating certain foods, you go to Greece and people are eating certain foods. And I think when we try to bring the whole world to be one diet, one food, I personally think with globalization and maybe putting McDonald's in every different areas of the world was not such a good idea, right? Putting Costco in every areas of the world may not have been such a good idea, because with globalization comes globesity, and then all these other diseases, autism, Alzheimer's, Parkinson's, COVID-19 around the world. I think if we left communities to stick to their microbes, to those regions, to improve the diversity of the planet, we would have a healthier planet. We would have a healthier planet with mental health. We would have a healthier planet with bowel problems, disease. But unfortunately, you can't escape this cross-contamination. And I think we need to be a little bit more selective of these microbes and say, these microbes, we don't need to eat tomatoes in December. We don't need to get these tomatoes from this other country and then therefore, infect our microbes, right? So I think that's how we have to see it. The one thing I learned from the farm was actually that 90% of the grains that our cows are fed come from other countries. And that boggles my mind, because there's a high rate of Mycobacteria paratuberculosis in other countries. Are we bringing those microbes in here, feeding our cows, and then the cows get a condition called Johne's disease, which is similar to Crohn's disease, right? And therefore, you're giving the beef to a kid, all of a sudden, no family history of Crohn's disease, and the kid has Crohn's disease. Is it really Crohn's, or is it Mycobacteria paratuberculosis that's in his gut that he got from the cow who ate the grains from another country? So, and then of course, that's why if you look at the protocols of Dr. Barodi with Crohn's disease, when you treat these kids with anti-MAP therapy, and then you give them fecal transplant, the kids are done with Crohn's disease. So there is an element of infection in Crohn's disease that we have to pay attention to. If you look at the studies of Yersinia enterolytica, was thought to be causing Crohn's disease, MAP, Malassezia furfur. I mean, so many microbes have been put in mice and then develop, those mice have symptoms like Crohn's, right? So I think there is an infectious component. I think there's probably also a genetic component. You inherit the gene, but are you inheriting the microbes as well? So I think the whole field is fascinating. And yes, it's going to open this hypothesis of mine.
I'm really excited, Sabine, that you are getting into the regenerative agriculture space. That's one of my passions, and also Adriana as well. So yeah, we integrate that a lot into how we talk with clients, how we teach clients, and are involved in that space, like promoting that to everyone, because it is crucial. It's not just the human microbiome, it's the earth microbiome.
Listen, all three of us joined for a reason, right? I mean, remember when you came in my office? What do we have in common? We're close to the earth, right?
Yeah.
We're into gardening. We're into everything I learned in GI, I learned from gardening. You have to pay attention to the herbs. You have to pay attention to the microbes and everything.
And I think we look similar as well. I don't know why, but I always thought
It's our microbes.
And I just realized the last time the three of us were together was 2 years ago.
I know. We need to get together again.
When your Malibu conference was. That was 2 years ago.
2 years. I know.
I wanted to hear if you wouldn't mind sharing more about what you are a pioneer in the microbiome research field in clinical trials around gut and gut diseases, and have been doing fecal matter transplants for a very long time and successfully treating people. So you have a really interesting perspective. Can you kind of tell us a little update on what you're doing in clinical trials?
Where we're at?
Where you're at, and then I realized you had shared that you're doing something with the Department of Defense too.
Yes. Yeah, well, that's part of my clinical trials. Yeah. So I'm GI, but I also do mostly clinical trials, and the Department of Defense is sponsoring a study on hospitalized patients. So they actually, I'm one of the physicians they pick to overlook those studies, because they know I'm not a person that is gonna, you can't buy me. Like, if the drug is good, it's gonna go to market. If the drug is bad, I'm a scientist at heart. So, and I'm a researcher, and I'm by the book. So, I like to sleep at night that the right drug went to market and that the wrong drug stayed off market. Because otherwise we're just drug dealers, right, as doctors. And I'm not here to be a drug dealer for a pharmaceutical company or anybody for that matter. I'm here to make sure that do no harm first, and to make sure the right drugs are going. So I was picked because, we've been working on clinical trials for pharmaceutical companies for a long time. And we've never had a 483. That's a fine, one of those fines that you get for findings, like for doing improper research. In fact, the FDA, finds all our charts, standard operating procedures by the book. We even had the European FDA come into our offices one time, and they stayed like 2 weeks in our offices, 4 agents. That was no picnic, but they didn't find anything. They thought everything we did was perfect. And, they looked at the drug critically. So, on the whole, I like to work with the agencies. I'm not scared of the agencies because I'm doing everything by the book. I like the fact that they're overlooking me. I like the fact that they actually are holding my hand for these fecal transplant, So because of clinical trials, I, got into the fecal transplant route, because whenever patients with C. diff couldn't get fixed, I would do fecal transplant on them. And then that opened up this whole world of, as the patient with Alzheimer's that remembered his daughter's date of birth, after 6 months of fecal transplant, was just an eye-opening for me, an eye-opener, and got me to speak to you, and then Dr. Feingold, and all that kind of brought those questions. I have so many questions. I'm always questioning. I'm questioning my existence. I'm questioning everything. So it opened the door to fecal transplant, but I needed to do the fecal transplant with the FDA, because, listen, even though they regulate, everything, they're still making sure that I'm doing no harm. So because otherwise we're just like, crazy scientists. We can't do that. You have to have multiple people looking at the research. So my research on fecal transplant is overlooked by the IRB regulatory boards, but also the FDA. I like the fact that they add more things that I didn't think about. And they also give me that extra layer of protection that if I'm doing fecal transplant on an autistic child, for example, which is one of the trials we got approved. So I got approved one that was metastatic mesothelioma. The patient lived way longer than she was expected. Unfortunately, she ended up back in the hospital. It was an interesting case because I gave her the microbiome of her grandson, and she started craving chicken nuggets just like her grandson. And she started eating, and she was having appetite, and she was having morale again and hope. And it was helping her with the chemotherapy. But then she ended up having a pneumonia, and then they gave her more antibiotics, and that killed whatever I did in the fecal transplant route. And I think that's part of the problem, is really, we need to continue doing these transplants if we want to see success, if we want to prolong life, right? And so, like any therapy, right? You don't just start a cholesterol pill and basically stop it. You continue it. You don't just start a diet and then you've lost the 10 pounds and then all of a sudden you're going to gain weight again. I mean, everything is consistency, right? You exercise for cardiac output, and you got to continue it. Otherwise, you're back to square one again. So I think consistency is one thing that we're discovering with fecal transplant. We need to do a lot more. The autism was approved, so I'm entering in that world, because when Dr. Adams' study came out, I got bombarded with families with autistic children who wanted me to do fecal transplant. So I wrote the protocol about probably 2 years ago almost, and we got approved finally. COVID hit, so we got stopped from doing it. So that's one of my projects that I'm involved with. We also at the same time submitted one for 30 patients. So we are hoping to get the green light from the FDA. It's, the first one passed, so now we're waiting for the 30. And of course, these are dangerous times to do fecal transplant, but my fecal transplant are familial FMT. So in other words, I take the donor. If I feel that the donor, like the sibling, is neurotypical and their microbiome is good and they match, and I see a problem of dysbiosis in the gut with the autistic child, then I feel comfortable putting that kid in a trial. And those are the families. So we're basically gathering right now the families that I feel like are the families that I would do fecal transplant on. What's beautiful about our technology, and I jumped into this, really wanting to understand. I think I was the first guinea pig, and we all know who else were guinea pigs, but I'm not going to say because of HIPAA. But it was beautiful to see the trillions of bugs we don't even know their existence, that gray zone. And then the 1% that we see, you see 10,000 bugs, bugs that you didn't even know were in your gut, right? Botulinum toxin, right? So
are you saying that you can see that because of your particular sequencer, right?
Yeah, so we analyze, so we have a particular sequencer that we got, and then we also developed an assay that is that allows us to go at a certain depth to see what we're seeing. And then we validated it. So in other words, we make sure that the microbes that we said we saw. And we verified it, we reproduced it, we showed, my stools, I can look at them now. I can give myself a hamburger tomorrow. I can eat a bunch of yogurts with, lactobacillus and bifidobacteria, and I'm still gonna have that same signature. I'm gonna recognize this as me. And that's amazing, because when you have a validated assay, you can from there on compare anything. You can eat a yogurt and see if it's beneficial without really changing the rest of your microbes. If your assay is not valid and it's not verified and it doesn't reproduce, and you don't really know what you're seeing, you're basically floating in space knowing nothing. But if you know and you understand these microbes, and Mars looks like this, and Mars is located at this, latitude and longitude, then you can find Mars again, right? And then you can kind of say, okay, well, this person has this microbe here. And I remember seeing it in this person. So when we started all this, because I'm in the clinical trial business, and none of that world of the microbiome made sense to me, because there was so much data that was conflicting and didn't make sense to GI doctors. We would be at meetings and say, what do you make of this lab test and this lab test? And they're like, I don't know. So I said, well, we need to figure it out. We're the doctors taking care of these patients with Crohn's and And if there's a truth to this, we need to see it. So I'm the first one to be open to everything, right? Somebody comes up to me and says, you know, this herb is great for anxiety. I'm going to say, yeah, show me the data. And then if they show me the data.
Show me the data.
And then if they show me the data, and my attending used to say that because I was one of those, you know, fellows in GI that was research fellows that would say, well, this is the way it is. And he'd be like, well, show me the data. And I'd have to bring him like 20 pages to convince him that I was right. And so it's the same thing here. Show me the data. Where does it say, show me the group that you took, you gave them beets, and the group you didn't give beets, and let's see who got better, you know, and who got worse. And that was the whole, you know, startup of whole placebo controlled trial, right? So in other words, you give beets to one person, you test them, and then you give the equivalent of beets that looks like beets to another person, you test them, and you see if there's data between them, right? And that's how research was thought of and validated and verified. And also when you can reproduce. So in other words, I'm taking beets and I'm giving them to Dr. Stolman in San Francisco. And I say, Neil, please verify that the beets are helping your patients with anxiety, right? And then all of a sudden he calls me, he's like, oh my God, you're right. Beets are helping. So that's reproducibility, right? So research has to be valid, has to be verified, and it has to be reproducible. If it's not those things, then we're basically selling crap. And, you know, there's crap and there's microbiome. That's totally different. So I think we have to be legit. And so to me, as I embarked in this microbiome world, and you saw me from the beginning, Jenny, I knew what I needed to get from the machines because Dr. Feingold told me. He guided me with all his protocols. He left me all his protocols, you know, the family did, and the books and everything. So to me, it was like a sign from God, like, take this on. And then from there, really, you know, I felt it needed to be done at the clinical trial level. Because if I'm going to say lactobacillus, and we've seen all these research from Japan and all these Zurich and all this, lactobacillus causes XYZ. Well, what does it do in the clinical level? Is it true? And then you can go back to your charts, you can go back to your patients, you can analyze everything and say, you know what? Yeah, they're right. I can validate. And that's what it needs to be. But unfortunately, so much of this research is not something that's a business, right? This is a passion project, right? It's like, I'm an astronaut, I want to discover space, and therefore I'm spending my money to discover space. That's what it is for me. It's a passion project. You know, what is vitamin D doing to my body? What is vitamin C doing to my body? You know, nobody's going to invest money in doing expensive genetic sequencing of the gut for vitamin C or vitamin D. Well, let me do it. I want to see. And, you know, so I started the nonprofit Microbiome Research Foundation because I felt, well, there's other people that would want to, you know, see this data. And maybe they'll sponsor the research. And, you know, even the research as we do it for autism, you know, there's people that would want to sponsor. So really, that's the beginning. And then, of course, the Malibu Microbiome meeting was set up with all of us as GI doctors to analyze, to see, to understand. For me to show them what I'm discovering in the microbiome, which is brand new to a lot of doctors, like a lot of my friends say to me, frankly, Sabine, I don't know what you're doing. And I literally say, I don't know what I'm doing either, but somehow I'm coming out with p-values of 0.0001, must be on the right path. So, you know, I always said, achieve a cure, understand the microbiome. And that was what it was all about for me. What is causing hair to grow after you do fecal transplant? What is causing a kid to improve with Crohn's disease after you give him a bunch of antibiotics and do fecal transplant? And it allowed me, that assay that we developed allowed me to show that data. And that's what got all these doctors to come on board in the GI meetings to see that data, to see what I was seeing. And that's what it's all about.
Yeah.
We're all curious finds.
What are you seeing in autism, Sabine? What are you seeing that works?
Well, for one thing, I did validate a little bit Dr. Rosie and Dr. Adams' data. We did see some kids with low Shannon index, low diversity. We did see some kids that had low bifidobacteria. We did see some kids that had low Prevotella, but we also saw some kids that had high Prevotella. So, you know, I think this is the big.
What about Clostridial species?
Yeah, so we did see some families where we saw a lower Clostridium species. I'll tell you what was fascinating to me is getting all the way to the species and finding the Sulfivibrio, which was a favorite of Dr. Feingold. And then in one family.
Is that just in one?
In one family, I found, no, actually I found it in 4 families, the Sulfivibrio.
The Sulfivibrio piger?
Yes. So we found it, so what happened is I was looking at the whole family and they looked all the same Shannon index, same everything. And then I started process of elimination. You know, you have to kind of do forensic medicine, right, with the microbiome. So I removed the bifidobacteria. Yes, he was low, but then so was the mom, but then it's explainable because the mom is stressed. She's dealing with an autistic child. So that wasn't really significant. And then I started going down the pipeline, Clostridium perfringens. We did find like E. ramosum, which has been, linked up with some inflammation of the gut. So that was exciting because we saw an elevation in the kid, but the rest of the family didn't. But then the Sulfivibrio was really significant. The kid had like 44,000 copies of the Sulfivibrio and the rest of the family had 0.
And so, that's a beta-glucuronidase species. So that would impair detoxification.
And so we're working with the mom with different products to try. So when you zone in on a microbe that you basically go, you know what, this may be the microbe, and Dr. Feingold found it in autistic children. Remember, we saw his freezer with all these, it breaks my heart.
You have those, right?
No, they're still at the VA. I'm still fighting with the VA to get them for me. They're like, well, it's proprietary to the VA. I'm gonna have to like go all the way to the White House to get those 2 samples.
Oh man.
I don't know, maybe working with the Department of Defense, I'll ask a favor. I need a favor. I want the stool samples.
Yeah,
Dr. Feingold.
I thought you had them. I don't know.
No, they never released them. So we have to recreate the whole library, but it's a shame because they had such a beautiful collection.
So Jenny and Sabine, can you tell our audience what are you girls talking about?
Dr. Feingold was very passionate about autism, right? And he believed that it was a gut dysbiosis and that it was actually a couple of microbes he thought were the cause. Clostridium perfringens, and then the other one, the Sulfivibrio. And so I'm always in tune and looking for those 2 because I always think, well, he published on it, maybe there's some truth to this, right? So, and that's what it's all about. That's my whole path is basically proving the giants. So hopefully I'm going to have a paper that's going to show, and I'm trying because with finding the Sulfivibrio, 44,000 copies of the Sulfivibrio, and actually proving that it's doing something is a different thing, right? So when you do genetic sequencing of the gut, it's not only, I found the Sulfivibrio DNA pipeline, right? So there's multiple pipelines we can do when we do genomic studies, right? We can look at the bacteria, we can look at the viruses, we could look at the fungi, we could look, and then we could look when we do microbiome studies, basically the metabolomes, right? The substances that are secreted by these bugs, right? So I think it's important. So the first part of the puzzle for me is obviously what is relatively abundant, right? So in other words, a kid has 44,000 copies of the Sulfivibrio in the gut. Is that relatively abundant? Is that the cause? I don't think you can say it's a cause. I think you have to say it's potential cause. We found other microbes that could be linked to autism in families. But I think more importantly, we need to see if those microbes are reproducing. In other words, run a messenger RNA pipeline to see, is it replicating? And because if it's replicating, then it's probably active, it's probably alive. And then the other one is basically looking at the metabolomes, looking at, you know, the secretions, the toxins of these bugs.
What are you doing to test the metabolome?
We're going to start doing that. So we're basically right now we're analyzing the relative abundance. Then we're doing the messenger RNA pipeline, and then we'll do so the beautiful thing is we collected the samples. So that's already, you know, we have a lot of collections. I think we have about 800 samples so far that we've collected. So that's amazing. So it gives us a look, right? And what's amazing, you know, is it gives us a look into the change. So a person had this microbiome at baseline and then they come in, they change. We know what's changed. So that's the beauty of that. You know, eventually once we have, you know, more funding, hopefully, you know, we'll bring in some, we'll start developing the metabolomic pipeline to look at those.
So
but
basically,
I think clinical picture is important. The relative abundance picture is important. I'm interested in fixing these kids. So to me, I think fixing the kids and seeing the change is going to tell me the story before the substances and the messenger RNA pipelines, etc. So understanding which microbe is at fault, trying different formulas, short of doing fecal transplant, and seeing if there's a change, you know, helps us advance the field, I think. And then it brings on other companies like pharmaceutical companies that are developing products. We're trying to get to pharmaceutical companies and say, look, you might as well step in and start looking at the microbiome now. Is your product, you know, hurting the microbiome? Because let me tell you, that's the motion that I'm putting in. I want vigilante. I want a vigilance in medicine, and I want a vigilance, you know, with these regulatory boards and the FDA to basically say we need to start judging, measuring better, looking at the quality better, oversight better, right? So that's what, you know, is pushing me in that direction. And I think, you know, it brings doctors to see my way and to follow. And I think that would be a win, really.
So basically, Sabine, what you said is you have seen different microbiome profiles in different autism people. So it's not always the same, but there is always a tendency to be imbalance. So there's always different types of overgrowths.
So there are some kids that we don't see anything. There are some families we don't see anything. And then one wonders if this is not a neurological problem first. You know, the kid, the 50% of Dr. Adams that did not succeed, one wonders if those 50% that didn't succeed, maybe it's because they had a neurological problem to begin with. And that's why it's important to have a global approach. So I'm working with Dr. Sheldon Jordan, who is the top, you know me, I like find the geniuses and I latch on to them and say, you're working with me, Dr. Barodi. I latched him on and, you know, we wrote the book together. And now I'm working with Dr. Yes, there you go. And now I'm working with Dr. Sheldon Jordan. So we're actually coming out with our book that's going to be coming out on neurological and the microbiome and incorporating. So I always joke, I say, I bring the microbiome, sh.t, he brings the brain. So, which is what it needs to, right? I mean, this guy is an interventional neurologist. He is the best of the best. If you go to his clinic, it's unbelievable. He's got top of the art, you know, from the blue machine to the ultrasound therapy to, you know, the nutrition pipeline to the MRIs that are specific to look at the brain. And he's interventional because he believes that he can fix Alzheimer's, Parkinson's, autism, etc. But it wasn't complete. And it wasn't complete until, you know, we look at other aspects, the nutritional aspect, the GI aspect, the rechanging the gut. And so that's why I'm excited to work. All my protocols, I'm working with him. We're writing, I wrote the autism protocol, had him joined up, and he's basically doing all the neurological workup for the kids to make sure, you know, all the testing are done appropriately, because we're going to need to show the FDA whether it works or not. And so we need a standardized test to show that it's working. So we have like 5 standardized testing that we're doing on these kids with neurological workup. And that's important, I think. That's part of the formula that needs to be done in the clinical trial before we start going into, well, let's start doing fecal capsules for all these kids. Because what happens if you cause harm and you kill a kid when you were trying to stop his autism, right? So I think it's important to have that relationship with the neurologist. So we're on the forefront, we're doing it. We hope to bring in other neurologists and other GI doctors to use our protocols in the future. We're going to write about it. So that's basically, we're stepping in also in Parkinson's, MS, Alzheimer's. So hopefully, you know, we'll see something.
And Sabine, talking about so you're testing the gut microbiome, and so you're going. Did I understand properly that you're going to start seeing the brain microbiome?
No, we're looking at the brain structure. So in other words, for Alzheimer's patients, we're doing MRIs. Yeah. That is looking at what's going on, what areas are you know, atrophied in the brain, where there's atrophy in the brain. I'm just simply focusing on the microbiome.
Mm-hmm. Perfect. And so how hard it is to, so you can, it's easy to see bacteria, and it's, I don't know if it's harder or easier to see viruses. How hard is to see fungus? And
so all those pipelines are different pipelines that you create in the lab. So in other words, you have a bacterial pipeline. So you have to do A, B, C, D, E, F, G. And then you have a viral pipeline, which is other reagents, other formulas, other ways to get to that. And then same thing with the fungus. I'll tell you, some of this work is 2 weeks to a month of work in a lab.
For one patient?
For one pipeline.
For one pipeline?
One pipeline for like 20 patients. So it's for COVID, for example, we looked at the first 8 patients to find COVID in the stools. We found the whole genome of COVID in the stools. It took my scientists 2.5 weeks and a lot of money on reagents to find to zoom in on the microbes and remove the bacteria, remove the viruses, and just focus on the COVID-19. So.
Wow. So
it's a lot of work.
Mm-hmm.
Is that work related to your new project with the Department of?
No, the Department of Health, the Department of Defense is just, they're testing a new medication for COVID in patients with the hospital.
Okay.
Yeah, but eventually, I mean, you know, I'm doing a fun study actually that I think you ladies would appreciate. I'm doing the microbiome of the farmers and their cow. So and it's actually the farmer, the wife, and the cow. So
okay.
So my Ginger, my manager, is said, Dr. Hazan, you're literally checking the farmer, the wife, and the cow. It should be like a song. So anyway, so we're checking it. We're giving them questionnaires. We're giving questionnaires to farmers all over the country. And basically we're asking them to fill in the questionnaire. They send them to us. We pick, you know, we pick accordingly. You know, we want to see whether regenerative farming is actually a good thing by looking at the microbiome of the farmer, the wife, and then the cow. And then we're going to be looking
versus conventional like monocrop.
Yeah, industrial farming.
Oh, okay, that's going to be really interesting.
Yes.
And then what I'm hoping is that's going to start a standardization to, you know, make the government pay attention and say, well, before you slaughter that cow and put the beef in all the butcheries and stores, maybe we should make sure the cow doesn't have tuberculosis. Maybe we should make sure the cow has diverse microbiome and is healthy. And so there's a joke going on in Facebook right now. Matt Clover is the farmer from Kansas. So there's Joel Salatin, who's in Virginia, I'm in California, and Matt is right in the middle in Kansas. And he's basically coordinating all the farmers. How did I meet Matt? So he said on the video and follow him on Facebook, he's hilarious. He called the page The Pharmacy, FARMACY, instead of medication. The Pharmacy. So anyways, so he called it that, and he basically said, may the best farmer win. And I said, win what? And he goes, I don't know what you give someone who has an amazing microbiome. So, my wheels, they already start turning. And I'm like, I know what you give them. You actually use them as donors, because that's where you want. You want the healthy donors, right? So basically, that's what he's doing to try to gather all the, how did I meet Matt Clover? It was an interesting story. His wife had COVID, and he spoke about it in a video with me. It was a YouTube video. It was the farmer and Dr. Hazan, and he basically said, Dr. Hazan, my wife had COVID, and I took her saliva, I smeared it on my face, I kissed her, I ate the foods with her, and I never got COVID. And then he goes, am I that worthless that COVID doesn't even want to come in my body? And then right off the bat, I was like, light bulb. I said, Matt, I want your stools. And actually, guess what? Amazing diversity. I mean, he's off the charts.
Wow.
So
can he be like a super donor?
Oh, he's gonna be so after I've evaluated him and everything, for sure, that's a super donor. So but that's the, I think, we have to start looking at what is making, but before even going to utilizing the microbiome of Matt, I think we should use, what is he eating? Mimic what he's eating. What he's doing,
eating, and what is in his soil,
and what's in his soil,
yes.
exactly.
In the air, even for the lungs.
Hey,
and also, like, if you listen to him and you follow him, the guy is pretty much like well balanced. First of all, he's very educated. He was a professor, so he's putting some science in his brain. And even though science is not his thing, he's educating himself. Amazing. He's a musician. He teaches the kids. So he's happy. He's in that music environment. He's in the farm. He's in the sun. He's in the air. He's eating right. It's a whole picture, right?
Yeah.
Yeah.
And that's what we want you ladies to teach the patients, because unfortunately the medical field is too rush. It's like insurances give the doctor 10 minutes with that patient. Here, 10 minutes, you're supposed to figure out everything. I mean, that's not going to happen.
Yeah.
I cannot possibly go over all the diets, all the traumas that person has had, all the complexities that made that disease in 10 minutes. It's ridiculous.
Yeah.
It's not a healthcare system. It's a sick care system.
And people should be getting upset with, people get upset with doctors for that.
Yeah.
But it's not the doctor's fault.
It's not.
It's not. I mean, there's anyways, I have a question.
It's not, and it's frustrating to the doctors because the doctors want to help, but literally they're in this box. They cannot go outside the box.
It's a really tight box. Yeah.
If you tell your patient to take XYZ, there's no guidelines that's protecting the doctors, and then the doctors are off. Could you imagine, like, if I started doing fecal transplant on all my patients, I'd be killed. I mean, that's why I have to do it with the FDA.
Mm-hmm.
Well, that's what we end up doing. We do a lot of education. We do that as health coaches. I do that certainly as a nurse because doctors don't have enough time. So I would say half my job is educating people about medication and their disease process and all of that. And it's the nurses that have to do it because there's no way. But I want to know, can I invite because my family are farmers, like
yes,
half of my family.
Invite them. Yes.
Okay. I would.
I'll send you the question. So go on the page,
Okay.
the pharmacy, and or Matt Clover. Well, you follow me on Facebook, so I'll connect to Matt.
Yeah.
I'll connect both of you to Matt. You really need to know him.
That is going to be so cool.
Yeah, we really want to collaborate with
yes,
the regenerative agriculture community, because it is going to be essential, just like
yes,
the microbiome.
essential.
Yeah, absolutely. Yeah. And so do you want to, is there any other research that you haven't talked about that we should know about?
So the data's coming on COVID-19 and the microbiome. So that's coming out.
Sneak peek about that?
No, I'm gonna be quiet.
Okay.
That's gonna be a surprise.
Diversity. I gave you diversity.
Diversity.
And by the way, diversity this is the movement I want you guys to like push and tell everybody in the naturopath world. Diversity is health. Loss of diversity is disease.
That's the first slide I show people when I'm doing a class, is the diversity slide.
Yes, and vice versa. Keeping humanity diversified. Yeah. We are all different. We need to remain different. We need to remain different in our cultures, in our foods that we're eating, in our behaviors. We need to remain different. The difference is where the beauty of humanity is, right? Yeah. So we shouldn't be looking at, oh my God, these people are off and these people, no, bring them in. The microbiome survives because it's all these microbes together. If you lose the Clostridiums or you lose the Bacteroides, you have a disease. That's it. That's that simple. So if we lose a culture, we've lost humanity. That's a portion of humanity that's disappearing.
And we need to tell people those are what we call keystone species. Like,
yes,
they are our protectors, our guardians, whatever you want to call them.
Yeah, and I think we have to maintain, we have to protect the Amazon jungle, but also protect the people in the Amazon jungle. We have to protect Central Africa. Central Africa doesn't have Alzheimer's, Parkinson's, autism. We're going to need their microbiome to readjust us, probably. We need to keep them, we need to preserve the microbiome of humanity. Because if we kill it with all these pesticides and all these fake probiotics and fake this and fake that, fake foods and fake everything, we're killing our microbiome. And when we kill our microbiome, humanity has disappeared. That's it. So it's just such a shame to me that, human beings have not been able to reproduce blood, right? Otherwise, we wouldn't need blood transfusion, right? So what makes a human being think that they can reproduce foods that are the same as what is given to us from the planet? A carrot is a carrot from the ground. The microbes of the ground made it. The microbes, the fertilizers, all that made that carrot. You can't make a rubber carrot in a lab. You can't make a fake strawberry. It all affects us. Yeah. And so these products, these foods that are just don't taste like food anymore. You go to Costco, you taste the strawberries. It doesn't even taste like strawberries.
Yeah. No, they taste like cardboard.
Yeah. And you have to ask, how come the strawberry from the farm that I know, like I go to my local farm in Malibu and it's amazing. It's sweet, it's tender. Tomatoes. You taste tomatoes in a grocery store. It is not the same as the tomatoes that you grew in your backyard, in your pots. So
Sabine, the last question is, so I do take probiotics, and my microbiome was terrible. I had lost all my bacteria. I have a test. I was in red, like the normal person was here and I was here. I was literally in red. So I was helped with probiotics, and I take different strains, but I take very good quality probiotics.
Yeah, that's a difference, right? So if you're taking good quality probiotics and you're noticing, wow, it's really helping me, then you're on the right path. That was the right probiotics for you. But the problem is that if you're, without looking at your gut and without looking at what is that doing to your own good bacteria, it's hard to say. You have symptom improvement, but not necessarily gut improvement without looking at the gut.
Yeah, and I hear what you're saying, because like this is what I do. I read labels. If I would say I do one thing, I read labels and study labels. So I notice that sometimes a client comes to me and they say, "Oh, I've been taking this probiotic, but I'm not sure if it's helping." So I said, "Okay, let me see what it has." And there is one strain. I'm like, okay, you're adding a strain to your microbiome, but that's
Right.
gonna do nothing to you, and who knows,
And you're lucky you actually found one strain, right? Because some
Yes.
probiotics we tested in our lab and we put them through the genetic pipeline. We found didn't even have the strains that were on the label. So
Mhm.
here you are reading a label and it doesn't even say what it's supposed to have. You know, that's the crazy thing part for me is who supervises that, right?
Mhm.
I don't think the FDA, well, definitely the FDA doesn't look at all the probiotics, okay? But even I know the FDA doesn't go to the actual vitamin or the actual, probiotic and actually retest them. There is no secondary check for that.
Mhm.
Needs to be.
Yeah. Excellent.
Oh, all right, guys. I'm gonna say goodbye because I got a lot.
It was really good to catch up with you.
Thank you. Same here.
Thank
I gotta
you.
lock up. Or
Thank you,
actually
Sabine, for
open
your
the door.
time.
Thank you. My pleasure. Bye, guys.
Bye. We just interviewed Dr. Sabine, the author of Let's Talk Shit. And this is Adriana Ponzanelli, and
I am Jenny Pandol.